All right, well, welcome, Maynard. I'm not sure if I'm supposed to say any remarks about this being part of a series. No? Okay. So, in thinking about things that we could talk about tonight, I was realizing that you were present at the start of 3 different departments: genetics at WashU, and MBT, and genome sciences. So they were all experiments in their own way, and you've had the pleasure to experience all of them. And then, of course, you were on the advance guard of the Human Genome Project and made major contributions to that. So we, and then there's the interplay between the science and the institutions. So I figure we have lots to talk about tonight. Let's start with MBT. Lee Hood clearly wanted to change the way departments were organized and teaching was done. And you were clearly attracted to that model in some way. What was it that got you interested in it?
So I have often joked that I seem to have about a 15-year attention span, and I think that my interest in MBT was just that the cycle was coming up. I'd been at WashU for 13 years and was kind of looking for something new to do. But yeah, MBT is a fascinating story, actually, and one that I'm not sure has been really told well. This audience here really comes at the genome sciences roots mostly from the genetics side, and so it might be fun just to talk a little bit about MBT.
Okay.
I think that so you asked how I was attracted to it, and that's a prospective question. What did it look like up front? And then there's a somewhat different issue as to how does it look retrospectively.
Yep.
And
Yep. I think we get one and then the other.
I think these are rather divergent in this case. I think, to me, the primary attraction was that it was obvious to anyone interested in sort of the formative idea of genomics, genome-wide approaches to genetics in the 80s, that it was technologically driven. It really wasn't biologically driven. It was technologically driven. We knew pretty much, what kinds of things we'd like to do. Exactly the things that are going on in this building right now. We just didn't know how to do any of them. And there were a lot of cultural obstacles to putting the technology in place. And so it appealed to me, the idea that there'd be this experiment to try to actually focus on pushing some of the component technologies so that of genomics kind of past the early startup phase and getting them a little more ready for prime time. And I think a lot of the sort of the rhetoric of the department really had to do with technology development in this more narrow sense. And I think that is an interesting topic, and it's relevant, of course, to genome sciences, where there's some of what I would call the sort of narrow form of technology development underway here, but it's certainly not the dominant activity, and turned out not to be the dominant activity in MBT. I think there's something about the academic environment that just doesn't lend itself to technology development in what I'm calling the rather narrow sense. That is, how to get better signal-to-noise ratio out of various types of devices, or the more engineering side of how you go from basically knowing how to do something in principle to being able to do it efficiently, cheaply, reliably, and so forth. If you look at the whole record of MBT actually had it was a remarkably successful experiment. Let's just focus on the actual science and technology that went on there, and the way that it fit or didn't fit into the institution is a sort of another narrative. But of the narrower kind of technology development, there were numerous projects that started there. The only one I can think of, I'm not sure we have any of our MBT veterans here, so I can really put my own spin on this. The only one I can think of that was unequivocal success, is a lot of people don't even know happened at UW, was a research scientist named Alan Blanchard, working off in a corner by himself, that probably many of you don't know, was sort of developing the integration of inkjet printer technology and chemical synthesis of oligonucleotides, and he was fussing with what solvents to use and what kinds of reaction times and what kinds of rinses and so forth, and actually got this working, and it really proved to be it was initially the basis, the whole basis, really technical basis of the Rosetta, which was eventually acquired by Merck, a fairly major sort of biotechnology success, because they really had a major competitive advantage for several years in being able to make customized arrays for expression studies. And it's still really, it is the antecedent of the whole Agilent system for making microarrays. So I think up front we sort of had the notion that a lot of this kind of thing would go on there, and actually didn't so much. If I were to identify,
so did it really? It didn't sound like it changed the cultural obstacles to doing technology development that you were talking about.
To a limited degree, it probably did, because I don't think an Alan Blanchard would have survived in most environments. Okay. He didn't really have a job. I don't know how he was supported. He just was there, and he was there for years. He didn't publish, and so forth. So there was some shifting of the culture. So that was a success. But if you look at the bigger successes, or the sort of larger scale successes of MBT, they were all highly integrative things that they actually didn't push the envelope, at least in my view, of any particular technology. They solved some problem that required bringing together an understanding of what it is you wanted to do, the necessary kind of state-of-the-art approaches. Not really a lot of fancy engineering. It was more the idea. So I could just give a couple of examples. I think maybe what I would rate as probably the three most successful kind of initiatives in MBT were John Yates's work, proteomics. So here the idea was pretty simple. I mean, there were people out there doing fancier things with mass specs and proteins and so forth, but the idea was just to really merge DNA sequencing, genomic sequencing with proteomics. And I actually remember, one of the absolute origins of this idea. Was a little sort of chalk talk thing that was given by actually a former undergraduate researcher in St. Louis in my lab, Andy Link. He's now a professor at Vanderbilt, who came through to give just a little talk about work he was doing in George Church's lab, and where he was a graduate student. And he'd found an unused Edmond degradation protein sequencer down the hall, and being a little eccentric, he didn't want to work on really nucleic acids the way everybody else was in George's lab. He wanted to work on proteins. So he sort of took over this sequencer and started cutting spots out of a 2D gel of E. coli proteins and putting them in this sequencer. And it didn't work very well. But the best ones were, some of the brighter spots were mixtures. You couldn't do 5,000 spot 2D electrophoresis and take these things you could only see with silver staining and put them in there. You needed a spot. And so he'd cut them out and put them in, and they were mixtures. And so he'd get these very complicated patterns coming out. They're mixtures of two or three things. And then he realized that wow, that's great. It's more bang for the buck. There was enough E. coli sequence in the database at that time. The genome sequence wasn't there, but there was quite a bit of sequence. And, a lot of the times he could figure out what the mixture was.
When was this?
So in 1992, something like that, maybe 3. And he gave this chalk talk, and there was a lot of discussion about how, that's what you ought to do. And so this was an environment in which, this kind of more integrative work, really did prosper. And other examples, like Phil's software, was certainly on a short list of major successes. And Phil can speak for himself, but I don't think he would claim that he solved any sort of computer science problems. Right. Obviously, he knew what he was doing, but he understood what actually was needed. And as you know from working in the field at that time, most people trying to develop this sort of software just didn't actually understand what the problem was.
Yes.
They could solve whatever problem they were working on. They didn't understand that basically the problem was to attach reliable quality scores to marginal quality bases, so that when you did assemblies, you could use 800 nucleotides of the read instead of truncating it at 250. And particularly in mammalian genomics, the human and the mouse, for example, a lot of the short interspersed repeat sequences are about 300 nucleotides long, and it just made a huge difference to be able to use all these data, but not be misled by it. You could use it to recognize the overlaps and so forth. So, you understood what the problem was, and I'm sure that requires that you be around a lot of people that are suffering over trying to assemble things that don't go together well and so forth. You just can't sit in your office and do these things. And I think Debbie's work on SNPs is still another example that was. Again, it was an integrative thing. People knew SNPs were important, but they just hadn't really gone out and figured out, how to really define what the kind of the SNP populations looked like in humans and so forth. So that was, I think, the kind of thing that worked well in MBT.
And what was it? What was it about? Was it the people that made it work like that, or was it the vision, the problem you were working on?
So it's always the people, of course, in the end. But I think one thing that, maybe if we run through our list of all these startups I've been involved in, part of it really had to do with just it was a new thing. I've always thrived in young departments. And I think and with genome sciences sort of facing its midlife crisis, 10th anniversary. It's interesting. I mean, why? A lot of good things happen in young departments.
We'll come back to that.
And so it was new. So people came here and they were going to do something new. They were like me. They were at the end of their attention span for whatever they've been doing before, and they were looking for something new to do. And they tried some of these more ambitious kinds of projects that were risky and so forth. And I think that's one of the interesting characteristics of startups, and something that is a challenge to sustain in mature departments.
So did it just come to the end of its cycle, or what didn't work?
What didn't work? Well, that's the other narrative. It's also relevant. It's not so much. I could tell a lot of anecdotes, but I'll pass on these.
We can cut the tape if we want.
It's also relevant if you abstract the question a little bit.
Yeah.
And it's relevant to any venture, any interdisciplinary science venture. Is that and any large-scale science venture, either of those. There's always kind of a tension between the research institute kind of vision that all of the overhead, literal and figurative, associated with the university. You got all these students around and all these rules, and you can't. It's a big kind of bureaucratic institution. And so it's always tempting. You see this today, you'll see it 10 years from now. There's always this temptation when you think you've got something that's very powerful going on to want to break off and kind of do it your own way without all of this kind of overhead.
I see. Yeah.
And then there's a more departmental tradition where the individual principal investigator, the kind of the young faculty members that are free to choose really what they do. They aren't expected the way they would be in a biotech company to get on some team thing that met institutional goals. This tension was there from the beginning in MBT and never went away. And really, genome sciences is the departmental kind of branch of that tension, and ISB is the more research institute branch. They're just two different ways of doing things. And I personally have always been a departmental creature. I just, departments can be irritating as hell, but to me, they work. And my good experiences in academics have always involved, being a member of a strong department that really works. Now, how to get some of that startup sort of energy and lack of definition and swing for the fences sort of mentality, without taking departments apart all the time and reforming them, that's a challenge. But in any event,
that's part of the cultural shift that you're talking about, though, that you started with, right?
yes,
That you have to, that the institution has to have some give on that, some relaxed expectations of some kind.
There's no question that the sort of business as usual approach to doing science is good at a lot of things, but it isn't associated with trying new things that run counter to the sort of received wisdom of how a molecular biologist or a geneticist or a computer scientist or somebody ought to operate. And just to talk briefly about St. Louis, I was telling Stan that if Bob and I start reminiscing too much about St. Louis, he should give me a sign. Get back to Seattle. Bob and I were next door to each other for at least 10 years in St. Louis, and this was a period when genomics, as we now know it, was really getting off the ground, and that certainly was a major center at which this happened. And that was the sort of, Don Schreffler, the founding chairman of that department, who hired both of us, he was the kind of anti-Lee Hood, is the sort of the other extreme. He was a very conservative Midwestern guy, just an absolute painstaking attention to detail. He worked, throughout most of his career on the mouse MHC, untangling, really difficult aspects of the genetics and so forth. But somehow or another, he created an environment there. He chose people and supported them, that might have had a little trouble in a more typical environment. I worked on the yeast map, for example, for I think 7 years before I published a paper, and it was a major activity in my lab. So you can't do this readily in any environment, but it worked there.
But you, when you interviewed at WashU, and I presume at other places, you gave your job talk on your tRNA work.
Yes. Yeah.
That was your cover.
Yeah. Well, right from the beginning. So the tRNA work, I started in Ben's lab, and it might be interesting just to really sort of briefly connect that to why I developed this kind of interest in doing whole genome analysis. So when I came to Ben's lab in 1974, really knowing nothing, one cannot exaggerate my ignorance at this point. Some people still have their questions, but in 1974 it was a clear-cut case.
I have to tell an anecdote here. When Maynard was, when we were hiring people in St. Louis, the standard was that they had to know more genetics than Maynard.
Right.
If they didn't pass that, we wouldn't hire them.
This turned out actually to be slightly more demanding than you might think. You know, there was certainly no one who knew less biology than I did, but you know, there was the occasional sort of biochemist or somatic cell geneticist, for example, who you know, just didn't meet the test. You know. But anyway, so I showed up in Ben's lab. You know, I was on sabbatical from Dartmouth College, where I was an assistant professor of chemistry. And this was real chemistry. We're not talking about biochemistry. I was an inorganic chemist and had no background at all in biology or biochemistry, for that matter. I'd never taken a college-level course in any of these areas. Still haven't. And so without going back sort of too far and trying to understand what aspect of my attention span had been exhausted, that led me to Ben's lab. My first 15 years were already up as a scientist and looking for something new to do. Anyway, there I was and started working on these tRNA genes, and you know, they were a good topic for somebody who wanted to learn a little basic kind of molecular biology of that time, because at least the gene product was relatively easy to purify and was well understood and accessible. And so I set to work purifying tyrosine tRNA. It had already been shown by biochemical genetic experiments that one class of nonsense suppressors in yeast inserted tyrosine at nonsense codons. Don Hawthorne had been the lead figure in the genetics of this system, and he was right upstairs and took sympathy on me on many occasions and explained some of the details to me. But in any event, we had these genes, and there was a fairly good genetic system about them, but the gene product was accessible. And going back to 1974, these things didn't all come together for hardly any system at that time. One or another of them could be true, but to find a family of genes that had good genetics associated with them and where the gene product was readily accessible, this is kind of what Janowski had been doing in E. coli, and Fred Sherman had been doing with cytochrome C and in yeast, but there weren't a lot of good cases. And so most people that were taking the general path that Ben and I were working on at that time worked on ribosomal genes because the gene product was readily available. It's a family, but there was no genetics, and we wanted to try to integrate with the genetics. Anyway, these genes were scattered around the genome. There were 8 of them, and all unlinked in the yeast genome, and all seemed to code for there was only one known tRNA, and in fact, as it turned out, they all have identical coding regions.
So at that point, were you isolating the tRNA itself?
Yes.
And that had been sequenced?
Yes, right.
Okay.
The tRNA had previously been purified, and the challenge was to purify the tyrosine tRNA and learn how to label it to high enough specific activity. It's just a little molecule, so that we could start to do RNA-DNA hybridization to kind of detect the genes. And that was my assignment. Ben wanted me to tell an anecdote or two about working with iodine-125 in his lab, our preferred labeling agent. It was, that was a different era. We bought both iodine-125 and P32 by the many tens of millicuries, and kind of did what we could with them. And I used to smuggle the isotopes across the international boundary north of here because it was the pre-9/11 era. And, Mike Smith and Gordon Tenner at UBC were important collaborators and just sources of help for us. And so I'd go up there and do some biochemistry and try to label these things and bring the stuff back across the border and so forth. You would be ill-advised today. Just to tell one more iodine-125 anecdote. We need a little variety here. Can't get too serious. My favorite one is, so then we later were collaborating with Ron Davis at Stanford, and I spent time there, and also was also took in my suitcase. I had 125 labeled tRNAs, but I got a little nervous about that because it was, there's a big sign at the airport. There weren't any guards or anything, but there was a big sign, about radioisotopes, and so I decided I was going to ship it legally, and this was cumbersome to say the least, but there was a way.
Yeah.
And so we got to the kind of the key moment, somewhere down in Health Sciences T-Wing somewhere. I had this appointment with the radiation safety guy to inspect the package, and there was a kind of shipping clerk representing whatever shipping company was going to transport this stuff, and so forth. And so I'd done all this pretty carefully. I was kind of nervous about it. I still don't do well with bureaucracies. Kind of nervous about this, and so there was not one photon coming out of this package. But I get down there, and the radiation safety guy takes out his Geiger counter and is screaming off scale, and so I don't know, what could I do? So well, take it back to the lab and I'll unpack it and so forth, but I don't understand this. That's all I could do. And so I go back to the lab and I start unpacking it, and there was not one photon coming out of this package. So then the lab phone rings, and this radiation safety guy said he found out what the problem was, is that the shipping clerk had been injected with 10 millicuries of iron 125 that morning. And I had about 50 microcuries in my little lead-sealed container. She could have gone out to SeaTac and got on any plane she wanted to and flown anywhere. So anyway, we were doing that. We were labeling tRNAs, and but the genes were all these unlinked genes. This really quite interested me, how how this worked, how the convergent evolution worked, and so forth. But mostly, I just got it. I was inherently involved in the genomics once we, with Ron Davis's help, got lambda cloning working. I could hybridize and get lots of lambda clones with random ends that, from all 8 of these loci, and we'd cut them with EcoR1 and look at their gel patterns, and could make contigs around all 8 of these loci, and it, was really a pretty direct step from there to say, really, that's what you should do to the whole genome is use shotgun methods and some simple fingerprinting thing, and let the n-squared part of the problem, comparing everything with everything else, be done in a computer instead of the way it was done then, which was by some complicated experimental procedure. So.
But well, I got a couple of questions for you here. I mean, first, what led you to pick Ben's lab?
So, Ben's lab. Yeah, well, sometimes, you're just lucky, and got to count on that. But that's kind of an interesting story. So I really didn't know anything about, I didn't know anybody working in molecular biology. There was nobody at Dartmouth. There was
Is that right?
Not one molecular biologist at Dartmouth. There were a couple of protein biochemists and so forth, but not a molecular biologist. I literally didn't know a molecular biologist. So I'd read a little bit about it, Watson's book, this kind of thing, and but I had no idea, what to do or where to go. But I knew I wanted to go and work in a lab, and so it went in a couple of stages. The first was
so you knew you wanted to work on DNA in some fashion.
Yeah, and I already liked the idea of getting it together with the genetics. This was clearly, when I read, for example, about Yanofsky's work, I thought that's fantastic. This is what you need to do. You need to do the biochemistry and do the genetics and somehow figure out how they relate to one another. And so I was looking for some place to go, and I had some weak family ties to Seattle, and so it was on a list of 10 places or so that my wife Betty and I just sort of talked about when I sort of got this sabbatical arranged. And then she wanted to pursue a master's degree in history during this sabbatical, and So she applied to a bunch of master's programs, and UW was one that she got into. And so that's why we came to UW, was so that she could work on her master's in history. I didn't know anything about the genetics department here, but I figured every major university would have some molecular biology. So then I started paying attention a little bit to the extent that I could to the University of Washington, and right I think the very week that we decided this, there was a paper in Science from Lee Hartwell's lab. There's a Brian Reed's kind of massive sort of description for a fairly general audience, a science audience, of the basic idea of the budding of the yeast and the cell cycle and the TS mutants and so forth, the epistasis tests. This is genetics I could actually understand, sort of. And it is illogical and so forth. And so there wasn't any molecular biology there, but it was a start. So I called up Lee, and he was very nice. He actually talked to me for quite a while. And
you were still in Dartmouth.
Yes,
yeah.
And to try to understand what my situation was, and he was the one who suggested that I call Ben. He thought we were kind of made for each other. Both had chemistry PhDs, and they both a little bit nuts, and just kind of anyway, it was a good pairing. And so that's how I ended up in Ben's lab.
Huh.
And so it went from tRNAs and iodinated this and that, and southerns and lambda cloning and so forth. And then I just could see that the yeast genome wasn't very big.
Well, it was still pretty big back then.
Yeah.
Lots of people were doing, not lots, but there were a few people doing just like you were doing in yeast, where they could get probes and so forth.
Yeah.
And over the next few years, that became a dominant industry.
Yes.
But not very many people were thinking about the whole genome. They were, was it that you weren't really interested in the particular genes, or?
Yeah, so that's an interesting and difficult question. I mean, it's certainly true. As you know, John Sulston and I were really the main early partisans of whole genome approaches, shotgun sampling, computational assembly, and so forth. And and we obviously came at it from about the polar opposites. Extremely sophisticated developmental geneticist. I mean, his Nobel Prize still is not for genomics, it's for developmental genetics. So he came at it from that point of view. Although, as John always likes to say, and this is certainly true, that he's basically a puzzle solver. And the way he always connects his work on the worm lineage to the genomics is they're both puzzles. He likes to solve puzzles, and I like to solve puzzles. And so
he also has a background in chemistry.
Yeah, and that may relate to how he got from there to doing developmental genetics. I don't know, but to a chemist, it's sort of a natural approach. And I often point, as far as the kind of conceptual focus, I always like to cite this chapter, which I'd already read at Dartmouth in the molecular biology of the gene. It disappeared after about the third edition when the book started to get too complicated. But the early editions of the molecular biology of the gene were laid out in a way that didn't require you to know much biology. And the third chapter was entitled "A Chemist's View of the Bacterial Cell." It's a brilliant essay. It's only about 10 pages. It's absolutely a brilliant essay in which Watson sort of talks about, how would a chemist look at a bacterial cell? I don't know what motivated him to write this chapter that way, but that's what he did. And so he talked a little bit about, what kind of molecules are in a cell. Well, you have small molecules like acetic acid that are in every kind of cell, and you have some medium-sized molecules like vitamin B12 and so forth, and we have all these macromolecules. That's what's there. And then he kind of went through, how do you, why is all this acetic acid? How does it get there? And showed some metabolic charts and how are these medium-sized molecules made, and decided they weren't interesting. They were just like acetic acid, but more steps. And but But then came the real tour de force. You can go and read this. I remember this was really just an absolute revelation to me. I think it was the first thing I'd ever heard said about biology that gave a little hope that you might actually be able to understand something. The chemists have a great skepticism, and that was what Watson was playing to in this article. He's very skeptical. He says, so gets to the end of the chapter and he says, well, this is a nice story that we've told, but how do we know it's even remotely true? We have these metabolic charts, that supposedly glucose, is metabolized like this and lysine is made like that. How do we know this is in the tip of some more or less infinite iceberg? Suppose that there are thousands of ways that glucose is synthesized and degraded in cells. How can you counter that claim? Well, actually, you can't. There's no biochemical argument. It just isn't one. Wasn't one then, and there isn't one today. Biochemists see the main stuff that's going on, and or they see exactly what they're looking for, and to prove that lots of other things aren't going on can't be done. And so I said, great, I've always wondered about that. And so he gets to the last page and he says, well, yeah, actually, can't be that bad. That he had a rough estimate of the genome size of E. coli from Cott curves, DNA re-annealing experiments. It was only off by about a factor of 2. And he knew that the genetic code was a non-overlapping triplet code. He didn't know about overlapping genes, but they're still not such a big factor. And so we just calculated, how many enzymes can E. coli have? And then he looked at all these metabolic charts and said, how many do we already know about? He said, wow, we already know about, 20% of them, 30% of them. It's not that bad. And I just thought that to me was just a fantastic insight. And that's genomics.
And that you read while you were still at Dartmouth?
Yes, right.
I see.
And it was only later that the full force of it as a kind of research program kind of hit me. But that's genomics. That's what Andy Link was doing with his spots. It's what we do today. We take advantage of the only thing, it's the only constraint on biological complexity. I think that remains the reason that genomics is powerful, is that biology doesn't have a lot of constraints. But this is one of them. It's not a very strong constraint, and so you read all these papers still today, that doesn't help that much because there's all this and that and the other thing going on, and the genome doesn't really tell you that much. It's missing the point completely. Sure, maybe we would like life to be simpler than it is, and for the constraints to be even stronger. But we got certainly, if you want to understand biology, you need to treasure, what constraints we've got. And this, I would argue, is the strongest constraint on the chemical complexity of cells. And so it's good to sort of reinvent biology around this constraint. And that's what's going on, basically. We should save some time to talk about the future.
We will. We have some, when it strikes midnight here, we won't turn into pumpkins either. So I want to talk a little bit more about WashU, because, as you said, you and John were pioneering the effort to describe genomes, not to a not very receptive audience, by and large.
Always a sign you're onto something.
And what was it about? I mean, you mentioned that WashU let you do this. What was it?
Yeah.
Were there particular things about the department, about the institution, that promoted this, or at least allowed it?
So
that other places wouldn't?
Yeah. So that is a hard question. I think that the geography is important. St. Louis lacks geographic competitors. It's constantly attempting to recruit graduate students, postdocs, and so forth whose sort of basic instincts are to go to coastal institutions. It's out there in the middle of nowhere, and and I think this does give rise to kind of opportunity that if you're trying to if a faculty recruiting search constantly comes down let's say in the 1980s to Tashnee's best postdoc and Paul Berg's best postdoc and so forth, which tends to be the case at say Yale. You get very good scientists this way, but you're not likely, to get someone who's going to go and work for 7 years on something that nobody thinks is a good idea without publishing a paper. It's just not what you get. And now, St. Louis probably didn't know quite what they were getting either, but I think there's a more tolerance in this type of environment, and more reliance on the sort of collegial instincts. That they're not. I never had this feeling. They were constantly soliciting letters. About whether I was in the top 11 or top 14% of some set of people. I had the feeling. People liked me there, and they were glad I was there, and that was good enough.
Yeah. And were there particular activities that either in Ben's lab or at WashU that were important?
Well, certainly, I sort of in Ben's lab, I kind of emphasized the how the genetics of the system kind of led toward my interest in a genome-wide approach. But also, I got a big dose in Ben's lab of how the techniques of molecular biology are getting better rapidly, and it's having a huge impact. And that you shouldn't always just ask the question, is this new thing right now, going to give me a new insight into the regulation of the globin gene or whatever? And that tends to be the culture of molecular biology is if some technical step is of immediate use to people.
Yeah.
RNAi was close to this model. There were a lot of people using RNAi before there was really any understanding of the mechanism, but they could knock down genes and see what the effects were. So that kind of technology takes off quickly. But technology that you can see is getting at the fundamentals of what we need to do, but doesn't work very well. It's hard to apply to the systems that many people are working on, so forth. This takes longer, and certainly in Ben's lab, that's what we did, and even in J-Wing, there were plenty of people that wondered, what if you compare the kind of genetics I was doing in Ben's lab with what was going on, let's say, in Lee's lab or numerous other labs in the department, there was no comparison. I had a few nonsense suppressors, and I was trying to figure out what genes coded for them, as opposed to trying to understand the cell cycle as a biochemical pathway. And certainly there were plenty of people who wonder, what are all these Southerns and Lambda clones and things? What are they doing for me? So I got a dose of that. I was just because during the time I was in Ben's lab, it was really the molecular biology as we still know it, kind of came together. Recombinant DNA methods, sort of Southern type analysis, RNA expression studies. All these things were kind of learned how to do them, and I think basically PCR is about the only major new player since I left Ben's lab as just a singular technique that is now everybody uses every day. But you could see this doesn't happen every decade. I'm interested in the history of science, and I could see that then. I was always interested in the history of science, and I could see that then. This is a really transitional period,
Yeah.
and so anyway, in St. Louis, I was determined to try to see how much mileage can we get computing, for example. I mean, now, how many computers are there in this building? I still am on your email lists down in Oregon, and half the email is about the cooling failing server room. As near as I can tell, they took the furnace out a long time ago. It's really just all these servers. And when I showed up I'd already been there a couple of years actually, because the first couple of years I was working on on the protocols, the lab protocols.
Yeah.
But so I needed a computer, and there were no computers in the department. There were none in our building. So I made a deal with an X-ray crystallographer that was about 3 buildings over, Len Banasak, that I'd buy him a new hard disk or something. It was $5,000, and held about, I don't know, 100K or something stuff. If I could use his VAX, timeshare on his VAX, and so we ran a wire, it went up those silos on the outside of the McDonald building and so forth, all the way from his lab over. Nobody had ever thought about how to run a wire from there. Why would you want to? And to the genetics department. Anyway, I got it up there, and I bought this terminal and brought it in one day, and it drew what in our little department would pass for a crowd. What's going on here? Maynard's really gone off the deep end this time. It's a genetics department. Now, there were some population geneticists, to be fair, in a different building that did some simulations of population, behavior of models, and they had some computers, but I had closer ties to the X-ray crystallographers. They were chemists, and anyway, so there I was with this computer, and I had to program it myself in Fortran and whatnot. But I did have confidence, this same confidence that I think was a real hallmark in Ben's lab, that these techniques are getting better. This is early days. We're not helping Phil Leader very much right now, but our time will come. But I think back to this thing about departments. I do think that it was a new department. We were all trying to really do new things, and I think that was certainly the case in MBT. That for all the problems we had, people were trying to do new things, and it's good to do new things. And there are a lot of disincentives, and they are worse now than they were then. I readily admit to young scientists, I don't give young scientists a lot of career advice, because the strategies I followed wouldn't work today. And that's just reality.
Okay. I do want to get to the future here. And as you mentioned, genome sciences is getting into middle age. This is
15 years is the real key.
I see.
That's when everybody's going to lose interest.
That's when everybody's going to lose interest. Okay. So, I mean, we are established. We maybe have one more, maybe 2 more positions to fill before this building is pretty well packed. We might be able to squeeze in, a little bit more, but basically, we're going to be in a different phase. We're going to grow by renew by replacement or something like that, and so, short of everybody jumping up and going to another place in 5 years, are there things that you see that the department could do to renew itself, to keep this energy?
No, those are hard questions. I mean, if there were a good solution to these problems, you'd see a lot of 50-year-old departments that have the same energy level as 10-year-old departments, and you find a lot of 50-year-old departments that are outstanding, but they, in general, just don't have the energy level. It's now I'm a kind of a Kuhnian, and Kuhn is an interesting figure in the philosophy of science, and it's not the time to address some of his more controversial ideas, but his simplest idea was to divide science into this kind of paradigm-shifting revolutionary science and normal science. That was his phrase, normal science, and he gives a good description of it. It's what it is, and he correctly points out that most of what we know about the universe has been learned during phases of normal science, because that's when we're working within some fairly well agreed upon kind of way to do things and learn things, and it works. And but what fun is that? And so the people with short attention spans, they're always looking for the next paradigm shift. And so how to favor that if you decide that that's what you want to do? I mean, this is great, I think, Stan's idea of having an ongoing kind of look at where you came from, where you might be headed this year is good. I don't think we spend enough time talking about just what are the big questions, what are the problems? Why can't we, why is it so hard to, you look at molecular biology and medicine, for example, broadly, not just genomics, but generally. Why is it so difficult to have a major impact? And well, you're not going to solve those problems by sitting around talking about them, but on the other hand, probably in American academics, there's not enough different forums in which people try to conceptualize what are the limiting factors. People are extremely busy doing what they've got to get done for the next day.
Yeah.
You've spent a lot of time in the UK, and had the old the MRC lab there had a big impact on your thinking, and I've watched one person after another kind of go there and spend a year or something, come back, or postdoc or whatever, come back and say, gee, we started having coffee, at 10 in the morning and tea at 2 in the afternoon, and stop what we're doing for a half hour or whatever it is, and sit around and talk. Not in our office, but go and talk. And so it's kind of pathetic, because the first week they're back, they get a teapot or something and put it on a little table like this, and say, gee, and 2 or 3 people come and listen to their stories about how much fun they had, and the next day there's only 1 or 2, and it's gone. It's never transplanted, and I don't have a ready solution, but I enjoyed your symposium. I was up this year for the symposium, and these were, obviously the speakers had been instructed to try to, really talk about what are the big issues they're struggling with. But I don't have an answer to that. I think it happens when it happens, and it tends to happen in young departments.
Okay, I'll maybe throw you an easier question. So I know you've been very interested in trying to get genomics into medicine, and how to do that. And I just wonder what you think how that is going to happen, where the future is going on that.
So that's a tough question, and of course
that's an easier one.
I should point out that I'm even less qualified to engage this subject than I was to label tRNAs in Ben's lab. Whatever one says about my biological education applies with even more force to medicine. A lot of the medicine, what I know about medicine, came from having lunch with Arno every couple of weeks for 10 years, and so I've had good tutoring, but don't want to. I come at the problem of how to have more effective interface between basic science and medicine very much from the basic science end of the spectrum. It is something that I have thought about a lot, and it's been a major activity of mine in the last year. I actually was involved in persuading the NIH to fund a National Research Council study on this issue, and was on the committee, whose report has just been issued, And during the last year, I spent quite a lot of time working on this study. And it's the worst possible time to be putting new policy initiatives on anybody's desk in Bethesda, where it's one of the periodic, except it's sort of the mother of all periodic, hunkered down sort of atmospheres there. How are they going to try to preserve what they're doing well as best they can, as opposed to how can we really confront the special opportunities that are in front of us. But the NIH and all of us should be asking the latter questions, because just look at the world we're living in. It is changing incredibly rapidly, and the whole healthcare system is in upheaval, and we haven't seen anything yet because we're still pretending that we can pay for the way we do things now. The integration of computers and healthcare is now happening. Everybody complains about it, but it is happening, and is transforming just the way that medical data are handled. And just look at the society. We went from all being unconnected, sort of little, kind of in our own little social groups, now to having this incredible level of connectivity. Concerns about privacy are changing rapidly. A lot of things are happening. But I would have to argue that the NIH, for example, is not showing any obvious response to this. They should be leading to take advantage of new opportunities. So that in any event, our report was pushing them in a particular direction. But I think just briefly that hype is not getting us very far, certainly with respect to sort of personalized medicine, or precision medicine, as we chose to call it in our report. Personalized medicine would seem to be a kind of oxymoron, because truly personalized medicine, there's no way of telling whether any treatment's better than any other treatment, because every individual has a different health history. We're going to have to group people, and in fact group them in significant size groups, or we're never going to make any progress in medicine. So we should stop talking about how we're going to somehow treat everyone differently. We would certainly like to treat them better and treat them more precisely.
Okay.
So why don't we pause there if you're...
So do you want to say more on that?
Yeah, just a little more about how to try to modernize. I think we should be thinking in terms of modernizing the interface between basic science and medicine. So what is the core problem that's needed to modernize this interface? Well, I think that it's difficult to exaggerate the amount of data that are going to be required. A good place to start would be to focus on 10 or 20 or 30 years from now, what kinds of data should a graduate student who's trying to understand, let's say, genotype-phenotype relationships in humans have access to, and how are we going to create data of that sort. Where they go from there, there should be some comments that underlies this enterprise, and the issue is how to build it and what should be in it. Where they go from there is going to depend on what they're working on. Sometimes they're going to go and do some experiments in mice or flies or yeast or do all sorts of things. But the core problem is going to require a lot of molecular data and long-term clinical data on at least low millions of people. I think it's relatively easy to calculate that that's just what you're going to need. We have some idea of what the complexity of the problem is, and some of the kinds of work that Jay and Debbie, for example, and others are doing in this department right now is every person poses a different set of clinical challenges, and every person's genome, poses a rather dramatically different set of kind of genotype-phenotype problems, and you can't untangle that issue. All this whole digression into common variant sort of thinking, first of all, never made the slightest amount of sense, and turns out to be wrong. it's really about idiosyncrasy and massive idiosyncrasy. So how do you deal with massive idiosyncrasy? It's a hard problem. Anyway, you're going to need a lot of data. So we should be thinking about building a database. Big biobank kinds of bureaucratized, super Framingham sorts of studies are, this is barely worked in the 20th century, and anyone who thinks that the 21st century is going to look like that, is really not reading the newspapers or just seeing how people behave now. It's completely the wrong model to think about pulling, a million people or millions of people in some sense sort of out of the healthcare system, and then spending hundreds of billions of dollars sort of doing something special with them in the hopes that decades from now this is going to somehow help. There have been proposals of this sort. They make no sense. What's required is that one step at a time we actually start to integrate more molecular surveillance with the ordinary course of medical care and the accumulation of electronic medical records about patients. It's going to have to be an opt-in system in which I think right now it is not a shortage of volunteers. It's a shortage of just any sense of how to organize such an effort. But I, in any event, think that for academics, the message is that a lot of what's being done now in genomics, for example, on this interface, is going to be a short-lived kind of activity. It actually makes very little sense to have the bulk of the kind of academic expertise in this area accumulating ad hoc data sets on this, that, and the other cohort of people that this, that, and the other thing about. We should be thinking about this problem more the way, for example, social scientists think about the census. So you can't do any kind of social science in a modern country without good census data. But we don't have big departments in the university with lots of people, hiring doorbell ringers and so forth to go out and get the census data. We assume that there is some system in place which provides just sort of basic data. And now the academics, play a critical role in analyzing these data, generating interesting ideas, testing them, developing new approaches to gathering the data and so forth. That's all fine. But the scale that this has to operate on is just not, and the nature of the activity is just not academic activity. And yet it's most of what's going on right now, I would argue, at the interface between genomics and medicine. That's what's going on in labs everywhere. That's what they do. And
but that's partly because none of it's being done in the healthcare system.
That's right.
Right.
So, whose problem is this? It's this is a classic policy issue. And so my argument, for example, to the NIH, is that that's phase one. This is not the most interesting part, but you've got to start somewhere. Is to shift these resources out of the kind of environment that they're largely parked in now, and put the leadership in the best organized of the HMOs, for example, that have big patient population bases, have good electronic medical records that are getting better. I think the gathering of molecular data is going to have to be paid for by the research enterprise because it doesn't have value added at the moment. Can't be justified in terms of its cost-effective impact on the patients. But it's getting cheaper, and so the NIH or other sources of research funding should be putting money in for that purpose, just to attach a lot more molecular data than we now have, and to build, potentially build fairly rapidly, that is over 10 or 20 years, this kind of information commons. And then the question is what in parallel with that, what kind of shifts are needed in graduate education, medical education, culture of the research culture and so forth. It's actually, I don't think it's so terribly complicated. I think we have to get out of what we're doing now, which is this sort of industrial strength kind of gathering of vast amounts of data that don't actually accumulate into an information commons. Actually, this department has ideal roots, and in thinking about the roots of this department, this is the roots you need. I think that this is what goes on in model organism genetics. It's not what goes on in most human genetics labs, but it's what goes on in model organism genetics. That's what people do. They assume that they can find the genotypic basis of, or at least the genotypic influences on whatever phenomenon they're studying. That's no longer, used to be the challenge, but it's not the challenge anymore. Then you have to set up some kind of system in which you can study the genotypic influences from from the genome all the way out to whatever biological endpoint you're studying. And so like the Genetic Society of America has had these meetings off and on, still having them. Really good idea. And this department's well situated to do this type of thing, is the sort of model organisms and human disease, something like that. This is exactly the right theme. There are discouragingly few examples in human genetics where you can really see how this needs to be done and how it can be done. And almost never do you want to develop this whole pathway purely in human systems anyway.
Yeah.
You're going to understand various parts of it in various other systems. So this is in a way, I was talking to Arno about this earlier, that I think genetics in general, over the time that I've been involved in it, because DNA was kind of the new thing and genomes became the new thing, there has been and still is an enormous emphasis on trying to figure out all this, what are all the genotypic kind of issues that relate to all the kinds of phenotypes. And humans, of course, pose a particularly daunting problem in this area, but that's what geneticists were doing. And that's really an anomaly in the history of genetics. We need to recover kind of the core mission of genetics, which is actually to understand kind of genotype-phenotype relationships in detail. That is how do causal changes in genomes kind of influence organisms. It's a hard problem. It's the only way forward in many of the problems in human genetics. If you've got highly idiosyncratic genomes, then you'd have to do what geneticists have always done. You've got to choose particularly interesting mutants and the people that are good at model organism genetics. It's not hard to get mutants. In yeast, a graduate student can get a lot of mutants in the second day that they're in the lab. The difference between a Lee Hartwell and a rotating graduate student, well, actually a lot of differences, but one of them is that he understands that the challenge is not getting mutants that muck up cell division. The problem is to figure out what mutants do you need. So in my big information commons, those mutants are going to be there, and the challenge is going to be to find them and then figure out what to do with them. And that briefly is where I think kind of human genetics is going to have to go to realize its potential.
Great. So let's open it up to questions. Ben.
To realize the vision you just gave us, do you think that concerns about patient confidentiality will be a barrier?
Yes, they are a big barrier.
Oh, Ben was asking if in the information commons that Maynard was talking about, would patient privacy concerns be a big barrier?
They are a big barrier, and it will be a central issue. We're going to have to find out how big a problem it is. As I said earlier, I mean, right now, the first 100,000 people would be easy to recruit. That's been shown. Neil Risch at Kaiser Northern California has signed up 100,000 participants in just that one HMO system. And the 100,000 was not limited by patient interest. It was limited just by how many people, interviewers and so forth, he could hire. And they have been happy to sign very broad informed consent. There were bigger difficulties negotiating these informed consent forms with several different IRB systems and so forth. But these are self-inflicted problems. They're not, and self-inflicted problems are, they're a problem, but they can change. The human subjects standards are going to have to change. HIPAA laws may have to change, but you don't start there. You deal with what you can with. First, we've got to deal with the self-inflicted problems before we get too concerned about statutory obstacles and so forth. Many, I think, the ELSI program associated with the Genome Project is one of the things I'm going this winter to be a visiting scholar at Duke, the Genome Institute there, and my main missions is to work with people there that are trying to figure out, what have been the lessons of the ELSI program, what have been the really useful lessons, and what are the problems that need more attention. So I think the most useful single thing to come out of the ELSI program, there's been a lot of problems, but there always are. The most interesting single thing, and it tended to be the philosophers that most forcefully and effectively pointed this out, is that we sort of tucked ourselves into what's come to be called a genetic exceptionalism frame of mind, as though genetics posed some truly powerfully unique kind of problems about privacy, ethics, and so forth. And there are, we all know, we're geneticists, we understand there are some things that are different about genetics than about various other types of medical data. But I'd be immensely more sensitive, just for example, just to take a common example from the ELSI literature, I'd be immensely more sensitive about whether my HIV status were generally known than anything in my genome. There are lots of medical tests that that we've worked with for a long time and have a lot of experience with that they're not genetic tests at all, that actually are highly predictive of people's health development and affect their insurability, their employability, their social acceptability, and so forth. And it's not that those aren't real problems, but they don't have associated with them this kind of platonic sort of idealism that surrounded genetic data. We just sort of persuaded ourselves that it was a uniquely dangerous kind of information. And so that the public is, from all indications, is in a more advanced position than a lot of the thinking in the human subjects community. There's still this idea that it's really dangerous. I doubt that that's the core problem. It's certainly a central issue, and it's going to need ongoing attention. But I think people understand that there's a basic compact in society that if you expect to have the benefits of increasing medical knowledge, you can't shield everything that involves you from any kind of scrutiny. And I believe these are manageable issues, and that they certainly, we shouldn't take the attitude that we have to solve them right up front. I think already we could recruit plenty of people into this endeavor. I would sign up in a minute, and and then we'll learn more as we go along. What are the real problems? I mean, we've had a large academic, quite a large academic enterprise out there now for 15 years, looking for really good examples of people whose lives have been badly compromised by the disclosure of genetic information about them. And it's really quite slim pickings compared to almost any other area. You know, people's financial status, many aspects of their personal lives, all sorts of things. It's easy to find examples of privacy breaches that have had a big impact on people, and you really have to look around hard to find examples where disclosure of genetic information has had that kind of effect. And so, anyway, I believe that we should bring genetics firmly. We should accept this sort of basic analysis that genetic exceptionalism was a mistaken doctrine. It's easy to understand how it arose, and I'm not trying to second guess the reasons that this happened. It had a lot to do with the history of eugenics and so forth. But you know, that was then, and this is now. We face a different set of challenges, so I think we can address them.
Celeste.
So it won't be long before every child that's born has its genome sequenced. How will medical doctors use that information? There seems to be a disconnect between the available technology and information and the ability to use that.
Yeah, there's a nearly complete disconnect.
Repeat it?
Oh, so the question was, Celeste had made the bold prediction, I guess it was a prediction, that it won't be long, you know, before every baby's genome gets sequenced and so forth. I'm a little skeptical of that premise, but let's work with it. And so the issue is, you know, what are you going to do with all this information? And well, very little, and that's why I'm actually skeptical it's actually going to happen, because the amount of medically actionable information there is really quite slight. You know, I wrote a news and views kind of thing about the Jim Watson genome in Nature a few years ago, and just one of the things I said in that article is that after looking at the data a bit, is that so if Jim sat down with a genetic counselor to sort of talk about the results of this test, it would actually be a very short conversation. There were about 3 recessive alleles that have population frequencies in the 1 in 10,000 range that might raise reproductive counseling issues, but you can do the calculation yourself as to how likely they would become an issue in his family's life. There wasn't anything there. There was nothing, and that would have any effect. And you go read Craig Venner's book. I haven't actually read it, but I'm pretty sure what you'd find in there are all these anecdotes, about how he's got this allele of that and this allele of that, and he's an insomniac and whatnot. And we just need to learn to call nonsense nonsense, and this is nonsense. There is absolutely no science behind all this. It's like astrology. And so, there's a big disconnect. We don't know what to do with this information. That's why we need data on, you can, of course, take the view, well, it's too hard.
No, that's my point, is that it's possible to get the data, but there's no way
to interpret it.
Right.
Yeah.
That's what's missing.
So, I think that what we need is a lot of molecular data on millions of patients that are followed over many decades. In this National Research Council report, one of the analogies that's in there, I wrote this myself, is that this is not going to be like the Apollo program or the Human Genome Project, some timeline-driven thing. We should think about this like building the European cathedrals. One generation is going to start them, the subsequent generations are going to finish them, plans will change repeatedly, the building's going to fall down every now and then. That's the way it's going to be, and we should be thinking about the problem more in that way, is that how, over the 21st century, can we maximize the chance that the actual human benefit that's there to be had, can be extracted. And I think, there is a huge disconnect, and the depth of the problem is not so widely understood. I mean, the New York Times in the last 2 weeks has had 2 articles about how it's all a computational problem, too much data and so forth. This has got absolutely nothing to do with computation. You all may not know how to cool a server room, but Google and Amazon and so forth do. Yeah, sure, it's going to take some CPU cycles, but this is not the problem. This has got nothing to do with the problem. Then you go to someone else and they say, and there are articles in the literature, $1,000 genome, $100,000 analysis. Well, anybody who's spent $100,000 having their genome analyzed, is a good person to sell, your underpriced condo to. It's not that it wouldn't cost $100,000; it's just the output would be useless. You wouldn't get anything for the $100,000. So it's not a problem that that there are a few people who actually know how to get out this information, but there aren't enough of them, and it costs a lot. It takes them a long time. We don't know how to do it. It's a scientific problem. We don't know how to interpret the information. So it needs to be looked at that way. There are other people who say, well, it's the conservatism of the medical community, and it's the way we train physicians. That's nonsense as well. The medical community, for whatever you say about it, I learned a fair amount about these guys, at least the academic medicine ones, my years in the Department of Medicine, and they actually have an outstanding record of taking up stuff that works. Got to help their patients. And then, it doesn't matter whether they learned about it in medical school or not. If they see it helping patients, then they'll learn about it and they'll use it. If anything, medicine has erred in the direction of taking up stuff that wasn't ready for prime time, as opposed to having really good diagnostic, therapeutic methods in the literature that they just don't use because they don't have time to look at it. The problem is it's a scientific problem, and it's a long-term scientific problem, and it's a fascinating problem, and we should get to work on it. And I think it will require some changes.
Joe.
It sounds like you're strongly backing the idea that common diseases won't be, as you said, caused by common alleles, but instead will be the result of a large number of rare alleles, potentially the large number of loci. Doesn't that also raise the prospect that many of these disorders, which we now call by a single name, will in fact turn out to be highly heterogeneous, and the model of finding the gene and designing the pill to cure the disorder is not going to be as easy to carry out. And maybe we'll have to go around telling people, no, there is no one thing called schizophrenia, or there is no one thing called autism, and we're going to have to divide these into a lot of much rarer disorders.
No, I mean, it's absolutely the case, and this. Oh, so the question was that,
Briefly.
That there's going to be a lot of heterogeneity, genetic heterogeneity in these diseases. There's going to be locus heterogeneity, allelic heterogeneity, and probably some other kinds that we don't have good names for yet. So it's going to be really heterogeneous. So how do you deal with that, basically? And that a lot of lumping has gone on in medical taxonomy. And that's all, I think undoubtedly true. And this report that I just have been working on this last year, along with a bunch of other people, is a bureaucratic enterprise, but is something like Toward Precision Medicine is the title. The subtitle is something about toward a new taxonomy, or something about a new taxonomy of disease. We need to think about how to classify diseases in a world in which there's going to be a huge amount of heterogeneity coming to the surface. And so some of it will just be finer subdivision. That'll be the easy part when it works, and we all know examples where it has worked. We used to have diabetes, now we have type 1 and type 2 diabetes, and we're on our way toward having many subtypes of particularly type 2 diabetes and so forth. Sometimes it'll work, we just need finer subdivisions, but the real problem is that it may not, a hierarchical system may start to just fail, and we don't have anything to replace it with. And so that's a good challenge. And there are undoubtedly better ways of doing it than anyone's thought of yet, but it also just may be hard. See, my point about the selection of the things that are worth studying reflects basically a view that I think that the biggest opportunities ahead do not involve better targeting of therapies that we already know about. That would be the sort of simple personalized medicine scenario, is that we have some big repertoire of powerful ways of intervening in disease, but we need to match those more accurately to a particular patient. I'm sure there are some opportunities there, but I'm actually reasonably impressed with how medicine, when it's at least practiced at its best, does that. Now, the biggest problem is that the cabinet is often empty. They just don't have good options. And I think a lot of what will come out of this world in which people get better at finding the rare case that's highly informative is that it will suggest interventions that will work, maybe not even work on that patient. It may just be informative about the pathology in that case in a way that suggests interventions that will work on some class of patients. That is, we will get new, highly effective treatments that we just don't have now. I think we're really not good at discovering novel, highly effective treatments. And I think that there are expanding opportunities in that area, but they're going to require kind of a restructuring of the way a lot of research is done.
Charles and then Arnold.
Maynard, you have a good record of this, choosing, quote, things that are worth studying. And I wanted to ask you, do you, at some stages I've heard of sort of a structure that you built into your life, especially when you were running the lab, that would be fun to hear about, number one. And number two, did this process begin when you were still a chemist?
Wow, so that, I suppose it's in the character of these events that the questions about when things did go well, what was the key to that? That's sort of the question. The first problem is, of course, as anybody who tries a lot of things will tell you, in the private sector and public sector anywhere, is that things don't usually go well. And so I wouldn't, I think I mentioned that I'm reluctant to give young scientists career advice now because I recognize that the environment has changed. And one way that it has changed that we all are complicit in, and that it probably can't be really rolled back. But you're going to fail a lot if you try to do things that are at all new. And I always got away with that. I made a lot of mistakes and did a lot of things that didn't work, but I got away with this. And so I was in environments where I, for one reason or another, could do that. We need to understand that I get tired of going to Bethesda and hearing that we're interested in high-risk proposals. They're really interested in high-risk proposals, providing that the probability of failure is near zero. You've got to choose one or the other. You're either interested in high-risk proposals or you're interested in things that are doomed to succeed, as one of my professors used to say. And we're doing way too much science today that's doomed to succeed. And we teach young people that it's a disaster. If you spend 2 years, 2 years, that's nothing. If you spend 2 years as a graduate student or a postdoc to accomplish anything. That if you can't point to numerous 2-year intervals, then you have nothing to show for it except maybe learning how to think a little more clearly. That you haven't taken enough risks. Anyway,
I wasn't asking it as advice for beginning scientists, but as a historical comment. Have you found special approaches to this? Did you go hide out for 6 weeks, or to think about important problems?
I suppose.
Repeat it?
Yeah. So it's just asking. Were there? Did I? Would I attribute any? Just the kind of things I got involved in to any particular habits of just managing my life and I spent my time. I've always been interested, and I think some of you know one of my retirement activities has been sort of looking at the interface between science and the humanities. And I've always been interested in the humanities. Spend a lot of time reading history, philosophy, literature. And I think that we train scientists too narrowly, and that I always tried to keep broad interests. And just it's a wonderful story. You just look at what humans have done in the last few hundred years. There have been some catastrophic errors. But if you look at it from an academic's point of view, I mean, how did the world look to us 500 years ago, and how does it look to us now? This is an astonishing story. I mean, this is just truly astonishing story. I've always been fascinated. How did it happen? Well, there's some things, that we can turn it into a more narrow history of science set of questions, which are amply interesting themselves. But it isn't all about that. It's probably not even mainly about that. It's about people, the way people thought, just about the lived experience of being a human changed, and changed dramatically. And I think that's where science is a human enterprise. And anyway, I always dabbled a lot in subjects that didn't have any very direct relevance to what I was doing.
Arno.
Well, a simple question. You were talking about this committee you can do work with. Is that available?
Yes.
To read something?
Yes, I'd be happy to send around you can download the PDF from NRC website, the full report.
And it's what's it called again?
Toward Precision Medicine is the.
Toward Precision Medicine. And it was just released in the last week or something? I went to the website a month ago or something and couldn't find
It
it.
was early November it was released. Yeah.
Okay.
By the National Research Council. And I was back in Bethesda, briefed Francis Collins and Harold Varmus and the usual suspects.
That's somewhat related to today, looking over the news, I came across something released by the NIH that they're going to spend millions of dollars, much of it goes here to this institution, on doing all kinds of genomics of all sorts. That if shots on, they're going to do more than ever has been done.
You're gonna read.
Is that an outcome already of the report?
No. So I think the question was, Arno was reading about NIH initiatives in sort of translational, genomically oriented translational research. And they're, if you go to Bethesda, they're very interested in high-risk proposals. They're also extremely interested in translational research proposals and so forth. I don't know in detail about the initiatives that you mentioned that are just being announced are definitely not influenced by our report. We'll be lucky if things that are happening 2 or 3 years from now are influenced by our report.
No, Debbie is going to get a lot of
Right.
And more
Debbie
power,
and Gail
more
both
power
are.
to her. Maybe Mark may know more about this than I do. it's not that there's not a lot going on. There's a lot going on the genotype-phenotype front.
And something else that's related to, they say, in fact, that the brain, medical where they want to use clinical tests that were done by genomic techniques and work these out in large amounts. And so Gail would be in charge of that, $7 million for that.
To get them into the clinic, you mean?
Yeah,
Does that
no,
mean?
to get clinical data for examining.
Yeah. No, there is, I mean, the NIH already is spending a lot of money on what I broadly would call genotype-phenotype issues. And my main point is that I'm always interested in the kind of the longer term, and so I don't have any strong opinion about what should be done this year or next year or the year after that. I believe that work of that type, that system for gathering information of that type, really needs to be modernized along the lines I described, and I'm skeptical that it should, let's say, on a time scale of 10 to 20 years, should be basically an academic enterprise. I think it should be more attached to HMOs and clinical chemistry laboratories and so forth. I just think that it's there's a big hurry. You go to Bethesda, you know, they're never interested in the long term. They're interested in getting, you know, something to happen, you know, now.
Well, one of the former PhD students is very much involved in that.
Oh, so Eric,
we should probably call it a night. Let you have some rest, and thank you very much for sharing your experiences
I
with
will
us.
leave
man.
Eric alone.